This is the recording. It's July 29th, lab, summer 2025, bio 105. All right, so here we are, Tuesday, okay? We're doing bacterial transformation today. You're gonna have a bacterial transformation quiz you gotta do. That quiz is going to require you make some predictions about the results that we're going to observe from the actual work we do today. Today is genetic engineering. We're going to be using a plasmid, which is an extra chromosomal piece of DNA that contains a gene for ampicillin resistance, as well as a gene for producing green fluorescent protein. So we're going to be very careful today in handling that plasmid since it encodes for antibiotic resistance. All right? So just be very cautious with that. You don't want to just, transform all the bacteria in your body to have ampicillin resistance. It's unlikely to happen, but, we don't want to take those odds. So gloves, goggles, safety, always. Okay. Tomorrow, we'll have our population genetics and Hardy-Weinberg dry lab, where I'm just going to be lecturing you on population genetics and Hardy-Weinberg equilibrium. There's an online assignment associated with this. Essentially, it's just another Hardy-Weinberg lab that you have to do online. It's only worth 10 points, though, because I find that a lot of students at this point in the semester will just choose not to do it. And I'm like, hey, it's 10 points then, You can take them or leave them, because it's a lot of work. Next week, we have one lab on gel electrophoresis, where we'll run your PCR products that you generated last week, as well as most likely some digests of the plasmid that we're working with this week. Then Tuesday of next week, we just look at our results from today's lab and prep for the lab final. So you can ask questions, I'll do example problems on the board, whatever. And then I will record that and publish it so that you can, use it as a review thing if you'd like to. Reasonable enough? Question? No? Okay. So you don't have to show up this Tuesday. But if you don't, and you fail the lab final, here's where my sympathy level's going to be for you. You know what I mean? All right. It's like, hey, we don't really have a lab activity on Tuesday. All right. Don't show up. It's fine. But you won't get the review session. All righty? Cool. Good deal. Any questions about what we've got left? Basically, we've got a week left of real classes for summer, and then we just hit lab final, lecture final to wrap it all up. Question?
The lab final is next Wednesday?
Next Wednesday.
Have, like, 3 more labs?
Yeah. We got today's lab, Wednesday's lab, next Monday's lab, and then the review session. So 3 real labs. Yeah, I know. 5 weeks. It's kind of a month has flown by, right? Yeah, we've spent a lot of hours in this room together. It's pretty nuts. Oh, thanks. Thank you. Any other questions about what we got ahead and coming up here? All right. Let's go ahead and look at Lab 9. Now, when I was looking at this right before class, I realized that this embedded video had broken, so I re-uploaded the embedded video, which has an additional demonstration of the transformation and answered some questions. It's a 7-minute long video. Gosh, we were having a hard time back then. I was, at least. Look at that, though. I figured out how to do video in video, frame in frame, or whatever they call that. It's pretty impressive, right? It's all synced up. No big deal. Well, it's not perfectly synced up, but whatever. So there's a little tutorial video for you right there if you want to see. Let's go ahead and do this thing. I've got a little PowerPoint that we can look at. about bacterial transformation. Where's my clicker? Okay. So, genetic transformation, what is it? It's when an organism takes up foreign DNA. We're doing bacterial transformation, so we're going to insert foreign DNA into E. coli. What's foreign DNA? It's DNA that wasn't inside you before. All right? Come on, you damn clicker. DNA is the genetic material. We know this. All right? Frederick Griffith, 1928 research, foundational research, indicating that sort of laid the groundwork for understanding that DNA is the molecule of inheritance. They had identified many different structures and molecules within cells, but scientists still disagreed on what molecule was actually serving for heritable characteristics. Everybody knew characteristics could be inherited. Animal husbandry, plant breeding, these things have been going on for thousands of years, right? Before there was a microscope. So everybody knew things could be inherited, but they didn't know what the molecule was. So we're going to talk a little bit more about all this research and how the genetic material was determined in lecture, okay? How DNA was determined to be the genetic material in lecture. But here, let's talk a tiny bit about this Griffith experiment, okay? So Griffith worked with Streptococcus pneumoniae, all right? The causal agent of pneumonia, hence the specific epithet right there. There are two strains of it. One is smooth, one is rough. The smooth one is pathogenic, the rough one is harmless. The smooth one has a protective polysaccharide coat. Now, this is a nice little reminder that genes don't have to directly encode everything. Think about it this way. The smooth coat is polysaccharide. DNA doesn't get transcribed or translated into a polysaccharide. It gets used to produce proteins. So how could a difference in genetic sequence lead to the production of a polysaccharide? I'll put it to you. How could the organism catalyze the production of a polysaccharide using proteins? Biological catalyst. Enzymes, people. So there's a metabolic pathway that makes this polysaccharide coating, right? And if you have the genes for making the enzymes that make the polysaccharide, now you can have the polysaccharide. So I just want to emphasize for you again that inheritance and genes, they don't always directly encode the thing. A lot of the molecules that get produced are produced because you have enzymes to produce them, not because you have a gene that produces the enzyme. Enzymes are just as important as all of the structural proteins and all the other things encoded by the genes. Okay? Anyway, smooth, protected, kills a mouse. Rough, not protected, mouse is fine. You take this thing, you inject it into a mouse, the mouse dies. It's terrible. Why would you do this? Thankfully, this is just a cartoon mouse, so it has no soul, so it's fine. All right? So here's the experiment. You inject these smooth cells, kills the mouse, right? You inject these rough cells, the mouse is fine. All right? You take the smooth cells that normally kill the mouse and you heat them up to kill them, the mouse is fine because those things are dead. But if you take the heat-killed pathogenic cells and living, all right, non-pathogenic cells and combine them, well, then the mouse dies. And then you extract from the mouse living S cells. So this doesn't really prove that DNA is the molecule of inheritance, but what it does prove is that something can be transferred between these organisms. there's something getting transferred from the dead cells to the living cells that gives them the capacity to become pathogenic. So Griffith discovers this and calls it transformation. All right? Change in genotype, change in phenotype. It's all because of the assimilation of foreign DNA, that being DNA that wasn't initially inside of the cell. All right? Cool. Foundational experiment, the Griffith experiment. Okay? It's one that I won't directly test you on, but you should be aware of because it just gets referenced, you know. So taking up foreign DNA. It's not a thing that like every cell can do, okay? Some cells are better at it than others. It could be beneficial for bacteria to be able to take up foreign DNA though, because let's say you have a population of 1,000 bacteria and one of them involves antibiotic resistance. Well, wouldn't you like to take up that DNA and become antibiotic resistant as well? Cells that can take up foreign DNA are what we call competent. There are some bacteria that are naturally competent. Some naturally competent cells include causal agents of things like pneumonia or gonorrhea or influenza. All right? Their competence allows them to pick up new pieces of DNA and to gain new powers. Daniel Burrell calls them like video game power-ups. You know, taking a class from him, he's in the chemistry department. And he'll be like, yeah, it's like a video game power-up. I'm like, it is, okay? Now, there are naturally competent cells, and you can make cells competent as well, all right? So there are a few ways of getting DNA into a cell. The cells that we're going to use, the E. coli that we use in today's protocol, have been made chemically competent. So what does that mean? Well, they've been exposed to chemicals that punch little holes in the cell, in the plasma membrane. That way, we're just going to dump some foreign DNA into a tube with them, and some of that DNA will go into the cell, just through diffusion. So these have been made chemically competent. You've punched some holes. Now think about DNA. What do we know about it? Well, it's got a net negative charge, and that's going to make it pretty hydrophilic, which means DNA is not normally going to freely cross the phospholipid bilayer, which means you have to do something to get the DNA across the phospholipid bilayer. Okay? So you can treat it with chemicals to make holes in the phospholipid bilayer, but that's going to hurt the bacteria. So then you have to be really nice to the bacteria and sort of nurse it back to health after you've transformed it. You can electroshock the cells. That makes little ruptures in the membrane too, little holes. It's called electroporation, all right, because you make little holes with electricity. You can also use microballistics, where you take the bacteria, you take a bunch of little tiny beads and you soak them in the DNA that you want to transform the bacteria with, and you use a little gun and you shoot the beads through the bacteria, and it'll pass through the bacteria, and some of them will survive, and the DNA will be stuck inside. These all have different rates of success and different applications, but they're all ways of doing this, you know? You can also transform things using bacterial intermediates, like using agrobacterium to get genes into a plant or something like that. Okay? There are tons of ways of getting foreign DNA into a cell. Sometimes it naturally occurs. Sometimes you help the process along, because normally it doesn't happen very rapidly. Okay, awesome. Transferring genes for antibiotic resistance between species, bad for us. Antibiotic resistance crisis. Plasmids. What are plasmids? Plasmids are extrachromosomal DNA. So if we're talking about a bacteria, all right, well then the chromosome is a single circular chromosome. We've talked about it briefly, but we've talked about a lot of things, right? Single chromosome, circular, all bundled up. Bacteria can also have extra chromosomal DNA. It's a little tiny circular piece of DNA that has genes on it. All right? These things get replicated on their own. They can be passed to other members of the same species or sometimes different species. It's pretty cool. And these little extra chromosomal pieces of DNA can have other genes on them, giving the bacterium new abilities, like resisting an antibiotic. Okay? Cool. Good, 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 good. All right. Plasmids are artificially engineered. Sometimes we want to get genes into bacteria. Either we're studying what they do, or they directly make something that we need, such as insulin. All right? We generate insulin with genetically transformed E. coli. And the insulin gene is jammed into them via a plasmid vector. All right? So, yeah. So if we're doing this transformation to get a gene into something, we want to make sure that it worked. Okay? Transformation efficiency is normally very low. So we're going to take like 10,000 E. coli cells today and try to transform them in each tube, and maybe 50 will actually take up the foreign DNA. All right? But that's enough as long as we have a way to kill all the bacteria that didn't take up the plasmid. So that requires inserting a gene like ampicillin resistance. Okay? So this is what it means, right? It's like you got 1,000, I think I just said it, 1,000 bacteria. You're trying to get them to make, in our case, green fluorescent protein. That's like our goal, is to get the bacteria to make that. The gene for making green fluorescent protein is on the same plasmid as the gene for ampicillin resistance. So if only 1 in 1,000 bacteria get that plasmid, they will be the only ones that can resist ampicillin. So then you just fill the chamber with poison, take the poison away, and the only thing surviving is the bacteria that can produce the thing that we want. So you use the same thing for like transforming things for insulin production. You transform as many bacteria as you can, then you hit them with ampicillin, that kills any that didn't get transformed. Then you give them some time to grow up again and, rebuild the colony. Usually you grow them on a growth media that has the antibiotic present, so only offspring that also have the plasmid persist. And you end up with a bunch of bacteria that all have the plasmid. Make sense? I love it. You also need to have an origin of replication. That's a specific region on the DNA that has where the DNA replication machinery can attach. We'll talk about that a lot more as we talk about DNA replication in the week 5 PowerPoint. Okay. Good. Plasmid maps. If you're using a plasmid, it'll come with a plasmid map. All right? A plasmid map tells us what genes are on there and where are they. And this can be very important if you're trying to use additional means to confirm that a transformation worked. All right? So here's an example of a plasmid. This plasmid has the LAC-Z gene on it. This is not the plasmid that we're using, so don't worry too much about what the genes are for. Here's a gene for ampicillin resistance. Here's oh my God, this is not the origin of replication. Is this the origin of replication? I think so. The point here is, check this out, right? Here's a plasmid map. Plasmids have a name that'll be in the center, and they have a length. This plasmid is 2,686 base pairs long. All right? Cool so far? A plasmid will also have genes on it. Here's the ampicillin resistance gene. The ampicillin resistance gene runs from base pair 2486 to base pair 1626. all right? So the length of the ampicillin resistance gene is 860 base pairs long in this case. That math work out? I think that's right. Cool. This is important. I promise it's important. Here's the plasmid that we're using, the P-Glow plasmid. Okay, it is 5,371 base pairs long, and here's what it has on it. It's got GFP sub UV. This means it has green fluorescent protein that will fluoresce under ultraviolet light. Okay, green fluorescent protein gene right here. It's under the control of the arabinose promoter. What this means is you have to have arabinose present for this protein to actually be produced. Normally in fall or spring, we would talk a lot more about this. I can give you more information if you're interested. We'll talk about it in lecture a little bit when we talk about bacterial operons and how genetic expression gets regulated. Okay, fair enough? Cool. It's got an origin of replication, which is necessary because it's got to replicate that plasmid. And then ampicillin resistant gene. Here's green fluorescent protein in action. Originally isolated from this jellyfish species. It's been mutated to fluoresce in the presence of ultraviolet light. Okay? And if bacteria transform, then it can be expressed. And the bacteria will glow under ultraviolet light. This was done in a lab like 4 years ago or something. You'll have an opportunity to take bacteria that you transform and draw a little picture with them if you want. Then we'll hit them with ultraviolet light and you'll be able to make it fluoresce. Okay, pretty cool. So glowfish, those have been genetically engineered to express green fluorescent or yellow fluorescent or red fluorescent protein. All right, so I gotta hit them with a special light to make them glow. Glow mouse, you can buy them if you want one. All right. Cool. We've got a protocol here. Do I have anything else exciting on this? I don't have anything else exciting on this. We're going to talk more about plasmid maps in the gel electrophoresis lab, which is next week. Okay, I think. Or it's tomorrow. Anybody remember? Next week? Sick. Thank you. I can't wait. We'll do a gel electrophoresis next week. And so we'll talk more about plasmid maps and things like that. Here's your protocol. It's pretty straightforward, okay? We've got these very expensive E. coli cells. Competent E. coli cells are one of the most expensive things that we'll be working with in the lab. They are very fragile because they've had a bunch of holes popped in them, right? They're chemically competent, remember that? So we have to store them at minus 80 degrees Celsius in a weird special glycerol solution because the glycerol solution will prevent, it doesn't crystallize in the same way, and so it will not shatter the cells. Now, getting to negative 80 degrees takes a lot of power and takes a special freezer. You call it a minus 80. And if you want to see a minus 80 freezer, you can come with me to acquire the E. coli cells today. It's just a freezer, but it's kind of cool. I don't know. You're totally welcome to come with me up there in a moment. Here's what we're going to do. We're going to take the E. coli cells. We basically just pipette in some plasmid. Squirt the plasmid in there, and now you got a tube that has E. coli and plasmid. Then you put the dang thing on ice. When you put it on ice, the plasmid and the E. coli cells, so dig this, right? We've been talking all about these cells, okay, the phospholipid bilayer, how they have a hydrophilic domain, the charged domain. If you cool everything down and there's not a bunch of energy, that negatively charged DNA will just go boop and stick onto the outside of the cell. Okay, so now it's right there. Then we blast it with heat and everything starts to wiggle, and sometimes, this is the inside, sometimes that plasmid will go bloop. Only like 1 in 1,000, 5 in 1,000, chance of that happening, but it's enough with the vast number of bacteria we have. So the free floating, cool it down, it sticks, heat it up again, it goes boop, like that. That's the sound it makes, very good. Then we just plate them onto some media. Some of the media has ampicillin in it, which is an antibiotic. Some of the media does not have ampicillin in it. Ampicillin is still an antibiotic, but you know, it feels like the sentence should conclude that way. Then you incubate them in our incubator. So we've got these plates here. Perfect. We've got laurel broth plates right here. Laurel broth is just a general nutrient mixture. So these plates are just good for growing bacteria, good for growing anything. Here we've got laurel broth and ampicillin plates. They've got ampicillin on them. It'll kill anything that doesn't have the plasmid, hopefully. Now we've done experiments in this lab before. I'm gonna set this down. This must be dizzying. I've done experiments in this lab before when we have extra laurel broth ampicillin plates, where I'll just like wipe a bench, you know, or take some pocket change and like press it onto the ampicillin plates. Ampicillin resistance is all around us. Maybe it's because of the rampant use of ampicillin resistance genes by scientists on this campus, or maybe it's just not that hard to be ampicillin resistant in this day and age. It's an old antibiotic, realistically. Yeah. Anyway, that's that. Great. So we're going to plate some onto media that has ampicillin and some that has no ampicillin. The plate that has ampicillin on it should only grow the bacteria that got transformed. The plate that has no ampicillin should grow all of the bacteria, including those that were not transformed. Any questions about that? Here are our expected results. E. coli plus piglo DNA tube, that should make colonies on the ampicillin plate. E. coli only with no piglo plasmid, they shouldn't be able to survive. Should have no life on these plates. These plates have no ampicillin, so you'll get what we call a lawn of bacteria, which is just a big sheet of bacterial growth. Cool? Question?
What's the third container?
Oh yeah, so dig it. That one is laurel broth, ampicillin, and arabinose. So the bacteria will grow on this plate, but they won't fluoresce until they're exposed to arabinose. It's really cool. If you want to regulate the expression of genes, you can engineer the vector with a specific promoter region. And then arabinose, check this out, right? Just bear with me for a second. This is fascinating. Okay. It's a short lab, no need to worry. Arabinose, right? It's a sugar. There are enzymes for breaking it down. But arabinose naturally only gets, or the enzymes, the arabinase enzymes, naturally only get produced if arabinose is present. So there's this little region that like promotes the expression of the genes. So what the genetic engineers have done is they've taken out the genes for arabinase and put in the green fluorescent protein gene. That way, this still controls green fluorescent protein expression, and you won't get any GFP unless you have arabinose present, which can be important. Sometimes you don't want the cells. We're going to be trying to treat them real nice today with like, we're going to put them into a little mixy hot tub situation where they can just hang out and regenerate, because we're going to shock them pretty drastically with this heat and this cold and all the chemicals that punched holes in their bilayers. So do we really want them to be making green fluorescent protein while they're trying to live? Kind of. So we'll streak a few plates of those, and then in the next couple of days, we'll streak from the transformed bacteria onto those plates in order to get actually glowing colonies. Long answer for your question, but that is the answer to your question. Anything else? Yes, please.
For today, we're not doing the third place.
We'll do a couple. Yeah, I just asked Ashley to make up, you know, a sleeve of them for us, so we'd have plenty. We'll do a few, you know, a couple, a few, several, and then save some for after we've confirmed transformation has worked. Any questions? Yes?
Are we so? Never mind. I don't know how to ask.
All right, well, hey, if it comes back to you, Just let me know. Here's actual pictures of what the plates looked like in a previous semester. Okay? LB ampicillin, LB ampiglo, LB and piglo, but no ampicillin. All right? Cool. In the old version of this lab, we would do a count of colonies. I decided it was kind of, not very fun. But if you want to count colonies, I'll show you how the colony counter works. It's just a magnifying glass with a light underneath it and a grid. Okay, makes it easier to count colonies on a plate. Okay, that's our transformation lab. It's kind of it's pretty straightforward protocol, honestly. Here it is, right here. It's got, you know how these protocols are now? It has all the background information that I've already talked to you about. There's a piglo plasmid map. Restriction enzymes we will talk about later. And yeah, bacterial transformation. Oh yeah. How do we actually, so I'll show you how to streak plates. And after we've got all of these things done. Once we've got the 1 hour incubation step going, okay? I've run this for 20 minutes and had it work just fine. We won't do the whole hour, okay? What we'll do is get to this step, then I'll do the whole training with you of how to set up your sterile workstation and how to streak a plate correctly and how to flame sterilize and how to use a spreader and blah, blah, blah. We have to use fire today, which is fun. It's fun to use fire. But we're also going to be especially careful. Okay. Everybody stoked? Everybody excited? All right, cool. We're almost done. Let's just keep high energy levels up and finish this lab. Okay. I'm going to end the recording. If you're watching this recording and you want to see the training of how to streak and do the loop, that's in the other video uploaded onto the module. Thank you. Okay.